Yeah, Guilhem. All right. So, hello. This will be quite different from the previous talk. By the way, I have changed the title and talk compared to what's announced in the abstract. I've chosen to narrow down a bit the topic given the available time. And this is entirely a work in progress, so this is all unpublished work. So compared to what we have seen during this week, so we have seen a lot of time series of community composition. So it can be about the time scale of a few hours, a few days, sometimes a few years in the case, for instance, of the human gut microbiota. And now I'd like to ask, what if we look at the dynamic of the microbiota over a much longer time scale, namely over the time scale, the host evolutionary time scale, in the case of a host-associated microbiota. So you may wonder whether it makes sense at all, given everything that's happened in the microbial community. So I try to argue that it may. For that, we need to look a bit at what happens in the assembly of the microbiota over shorter timescales. So over one generation, so starting from the parental generation, you have vertical transmission of some of the microbes from parental to the new generation. And this can sustain very long-term persistence of some microbe along over many generations. But then you also have some part of the microbiota that comes from horizontal transfer from other hosts, and also that is acquired from the environment. And this horizontal transfer often comes from hosts that are nearby and so that are related to the parental host. And environmental acquisition comes from an environment that is often also conserved over long time scales due to habitat, to the niche of the host. And so all these different processes lead to, over many host generations, to phylosymbiosis, namely more closely related species, host species tend to have more similar microbiota composition. So the problem is that we don't have time series of this, obviously. We only can observe the microbiota at present time. And one of the motivations for this work is that over the recent years, there's been more and more available microbiota for many different host species. For instance, here you have more than 200 species of mammals spread across the phylogenetic tree of mammals. And the goal here is, thanks to some modeling, to ask some question about, to answer to some question about the history of this microbiota, namely to what extent the evolution of the host influences its microbiota, how are the different microbes correlating in abundance across evolutionary time, and what is the ancestral microbiota composition at nodes in the phylogenetic tree. So to do that, we built a simple model that is directly inspired from what is done in phylogenetic comparative methods, so in macroevolution, to model the trait of a host over its phylogenetic history. So the trait of any species over their phylogenetic tree. Except that here we're applying it to the composition of the gut microbiota. And so we're modeling the log abundance of the absolute abundances as a multivariate Brownian motion along the tree. And so this starts from some initial abundances, and it's parameterized by a variance-covariance matrix between the absolute log abundances. An important aspect here is that we don't observe the absolute abundances. So we actually have to make them latent variables and that are not directly observed. And this makes inference more complicated. We have to resort to Bayesian MCMC instead of doing some more simple maximum likelihood inference. And then a key feature to build into the model is to be able to distinguish between the part of the variation in the microbiota across species that is related to this evolutionary history of the host and the part that isn't. Because obviously there are many other reasons why there might be difference between different species in their microbiota. And for that, we use a well-known trick from phylogenetic comparative methods, which is Pagels lambda transformation. And we basically compress the tree. So we basically elongate the branches toward the present to account for the parts of the microbiota composition that has evolved independently in the different host species. And so this is parameterized by parameter lambda. So if we are lambda equal 1, it means that it can be fully explained by a Brennan motion along the phylogenetic tree of the host. And if lambda equals 0 means that it's just independent evolution without any phylosymbiosis. You can also add an additional layer of variation at present with a sampling step that can also account for the fact that for a given species you also have different variation among across individuals, but I don't want to talk about it here. And so now let's apply this to the phylogenetic tree of mammals that I just showed you at the level of bacterial order. So here are the microbes, the taxonomic units we're considering are the bacterial orders and their relative abundance in each host species in 215 mammal species. And we find lambda equals 0.65, so it's a relatively high phylosymbiosis. Actually, you can see it by high that there are some phylogenetic structure in the composition. But you also did it for birds, and this is more tricky because birds are known to have very, actually it's often argued in recent literature that there is no phylosymbiosis in birds. We find that it's indeed lower, half lower, but it's not. There is indeed there is phylosymbiosis in birds, although it's not as obvious if you look at the microbiota composition at present. And so why? So you may wonder why. So whether this signal we're finding might not be confounded by the fact that some of the covariates at present that may explain the microbiota composition, such as diet typically, may also be phylogenetically structured. And so we're currently trying to build this into the model to try to disentangle the effect of such covariates. But what we did so far, very simply, is to shuffle the species within each category, for instance, diet, and rerun the model many times. And it's a way to assess whether you find some phylogenetic signal once you have broken the phylogenetic structure within covariate category, such as diet category typically. And when we do that, we find that, yes, there is some signal that can be explained by diet. Realm is here is a biogeographic unit, so it's a way to account for different type of environment at the global scale. But it doesn't explain all of it by far. And flight is the fact that I mean it refers to the fact that there is actually I mean, as you could see on the previous slide, bats have a strongly different microbiota, and there has been some hypothesis made in the literature that this might have some. So this may explain some of the variation in microbiota because of the constraints on the gut microbiota that are imposed by flight that make the intestinal tract shorter, and that it doesn't allow the host to control its microbiota as well. So that does explain some part of the composition, but not nearly all of it. And in the birds, basically it's explained nothing to add this covariate. So we can then look at, if you remember the model, we have this covariance matrix between the different microbes that is inferred. And so it's the covariance matrix between the abundance of the different microbes along time. And so here I show it for mammals and birds. And what you can see is that the pattern is fairly similar between the mammals and birds, which means that basically the bacterial order that tend to correlate across time in mammals also tend to correlate across birds, and it's very easy to see if we plot the covariance matrix entries of mammals versus birds. On the original plot, I was also showing the ancestral microbiota composition at the nodes of the tree. And so if we look at the ancestral microbiota composition at the base of all mammals and at the base of all birds, this is what we find. So there's limits to this exercise in terms of how we can really interpret it. But it's interesting to look at how this microbiota falls on the PCA of the different present-day microbiota, and we can here plot them according to the diet. And well, on birds, not too unexpectedly, there is not much structure, but on mammals, you can see that the herbivores onto the right, carnivore to the bottom right, and in the middle, you have a big cloud that corresponds to the invertebrate eaters, so mostly insectivorous, and you can see that the ancestral, so the blue dots, which is the ancestral microbiota of all mammals, it kind of falls in near the clouds of invertebrate eater. So that's the nearest species in that PCI space, and it's interesting to see that it fits with the assumption about the diet of the ancestor of all mammals, as far as it is currently assumed. So what we found is that there is phylosymbiosis in both birds, mammals and bird microbiota, and that it's much higher in mammals than in birds. So microbiologists have also run this model on a compiled dataset of vascular plant root microbiota at another point, and they found very low phylosymbiosis. And from those 3 points, we can make the hypothesis. So it's a hypothesis that has been made, but it's the first time it's confirmed in a model-based way. So that basically you have a gradient of phylosymbiosis that corresponds to how much the microbiota is, so how much the host can exert control over its microbiota from the ecosystem on the leash in mammals, where the host exerts tight control over its microbiota, and there is limited impact of the interaction with the environment, to the root microbiota of plants, which is very different. So open system, a strong interaction with the environment, a strong exchange between different hosts, and also a stronger control of the microbiota themselves on the host through symbiotic associations. So to conclude, we've shown that host evolution has a significant impact on microbiota composition in mammals and birds. Diet and biogeography conservatisms are not sufficient to explain those patterns we find in mammals and birds. The abundances of the different bacterial orders correlate between each other through evolutionary time in a way that is similar in birds and mammals. And we can look at, this model gives insights into the potential ancestral microbiota composition at the base of the mammal and bird trees. And finally, we can conclude based on these results. These results support the hypothesis that the strengths of the phylosymbiosis primarily depend on the strengths of the host control over its microbiota. Thank you.
Thanks for keeping to the time. We got time for questions. Chris?
Oh yeah, that's super fascinating.
Thank you.
Random fact question. So is there any difference between fruit bats and insectivorous bats?
Actually, well, I have not looked at this specific point, but fruit bats are classified as fruit eaters. So they're not classified in our randomization. They're not classified as obviously as insectivorous. So I mean, this is accounted for, but I have not looked at the specific difference.
Well, that same question, the length of the alimentary canal, covariates rather than flight.
Yeah, this would be better. This would be more mechanistic for sure, but this, I mean, it's not so easy to obtain that information for all the species, I think. Yeah. Can go to the Natural History Museum in London, I guess.
All right. More questions? All right. If not, let's thank our speaker again.